A very good day, Doctor, and to my fellow classmates. Today we'll be presenting about egg collection of bluefin tuna. My name is Adriana, Shazreen, and these are my fellow group members. Today we'll be focusing on the process of egg collection, fertilizing and hatching rate of the eggs, stages of egg development, and also the importance of egg collection. But firstly, what is egg collection? Egg collection is a method of collecting fertilized eggs. The method depends on the type of eggs that is produced. For bluefin tuna, they produce floating eggs. The eggs will float until hatching time. In culture system, it is best to collect the eggs as soon as possible to avoid the eggs from entering the filters. Nets and pipes can be used to collect the eggs. Atlantic bluefin tuna eggs float at a certain distance from the ocean surface, and they are at the mercy of water currents. Each egg produced is 1 mm, and when they hatch, they are 3 mm.
Hello, my name is Riyadh, and I will be presenting about the process of egg collecting in Atlantic bluefin tuna. So I will be explaining about two methods that are commonly used for egg collection in Atlantic bluefin tuna. For the first method, the unfertilized eggs or sperm are obtained from a sexually matured bluefin tuna by massaging the belly and letting the eggs or sperm fall into a dry container. In the laboratory, the eggs and sperm are then mixed and seawater are poured into it to activate them. And the fertilized egg are then kept in laboratory until it hatch into fry. Now for the second method, the day after the hormonal induction, a blue PVC curtain are attached to the net within the cage to contain the egg. Then manual egg collection are executed by using a 500 micrometer plankton net and then gently transferred into a bucket containing oxygenated seawater. And eggs that are further towards the center of the cage are reached by rubber dinghy or a small boat.
Hello everyone. So this is the second subtopic for this topic, which is the fertilization rate and hatching rate of Atlantic bluefin tuna, ABFT. So I'll start for the fertilization rate, which is ABFT is depending on only two things, which is sperm quality and mating behavior. If the temperature is a good quality, suitable condition, and the water wave is low, they will be mating better. Does it? Although most Luna species exhibit particular mating behavior with male chasing the female, external fecundation, and the very large number of expelled gametes make fertilization rate of fishes, which is ABFT, difficult to measure. And the second one is hatching rates. Through my explanation, previous explanation last week is they will hatching only 80%. Only 80% in 24 to 48 hours only, and the rest will be not in a good quality. Then the best thing is they will be doing normal hatching without any lethal malformation between 40 to 60%. That's all from me. Thank you.
Hello, I am Nurul Nasuha, and today I'm going to talk about the egg development stages of Atlantic bluefin tuna. According to Abbascal and Medina 2005, there is 5 stages of egg development, which are early oocyte and folliculogenesis, primary oocyte growth, the lipid stage, the vitellogenesis stage, and then the last stage is maturation stage. In the first stage, the oocyte diameter is about 10 to 20 micrometer. The nucleus has a single and central nucleus, whereas the modest amount of cytoplasm contains many ribosomes. After that, the oocyte diameter increased to 20 to 120 micrometer in the second stage. The cytoplasm is highly granular due to an abundance of ribosomes, and then the volume of the oocytes increase as a result of proliferation of membranous organelles and accumulation of ribonucleoprotein in the cytoplasm. In the lipid stage, the oocyte diameter is about 120 to 250 micrometer, which the beginning of this stage is marked by the appearance of lipid droplets that can be distinctly identified on light microscopy. The lipid synthesis continues throughout this period and extends into the vitellogenic stage. During the vitellogenic stage, the oocyte's diameter is about 250 to 500 micrometer, in which the oocytes accumulate membrane-bound yolk platelets that increase in size as the oocyte development progresses. Also, the vitellogenic oocytes have abundant finger-like projections that are embedded in the layered vitelline envelope. For the last stage, the oocyte diameter is about 500 to 1000 micrometer, which is also known for about 1 millimeter. And then the primary event of oocyte maturation is the resumption of meiosis, the migration of the nucleus toward the animal pole of the oocytes, which is followed by the breakdown of the nuclear envelope. For the lipid droplets, it is begin to fuse into larger lipid inclusions that occupy the central portion of the oocytes, where they eventually form a very large central lipid droplet. Thank you.
Hi, my name is Alisha, and now I will talk about the importance of egg collection. Number 1 is to observe and examine the stages of egg development for further research. Now, this is very important because in order for us to understand the biological behavior of fish, we need to study up until the stages of egg development, because fish are very fragile at their egg development stage, so we need to take extra care for us to be able to produce a good quality of fish. Next is to prevent damage on fertilized eggs, which can reduce the percentage of hatching rate in eggs. If we fail to follow the correct procedure in collecting eggs, we might decrease the hatching rate of eggs, which will then affect the survival rate of fish. If we follow the correct procedure in collecting eggs, and we also do a lot of practices, we are used to it, especially for labor work, we can ensure good timing and efficiency in egg collection, which is a very good point, because this can also increase the survival rate of fish in their further stages of development. And lastly, through multiple practices of egg collection technique, we can become familiar with the spawning patterns of cultured species. Because if we understand the spawning patterns of the fish, we are able to be ready whenever we need to set up the egg collection method, so that it will not affect the survival rate, the hatching rate, or even the fertilization rate of eggs. In conclusion, egg collection is one of the most important methods in aquaculture sector. Factors such as the size of cage, water flow rates, and even labor work, especially in pelagic eggs, are some of the few factors which need to be taken into consideration when collecting eggs. Frankly, an efficient procedure of egg collection is the key of success of an aquaculture operation. And that is all from us. Thank you.